So before we start, hands up if anyone's ever overwatered a houseplant and then gotten so many flies on it. Yes. So those flies are fungus gnats, and unfortunately, I'm not here to solve this problem for you. However, instead, I can help you maybe understand why these flies are so cool. And because basically they've got really weird genetics and a really weird sex-determining system. And basically understand how studying these flies on houseplants can offer us some solutions to limiting their impact on agriculture, such as the mushroom farms that we have. So let's dive in. Fungus gnats. I'll first explain what normal genetics will look like, then what fungus gnat genetics looks like and why it's so odd. Then I'll outline what my research actually is, and then implications of it. So, normal genetics. In terms of sexual reproduction in organisms, the rhetoric that most of us would have learned in biology class is that a child receives one copy of each chromosome, the molecule that encodes genetic data from each parent. Then they pass on these chromosomes to their own child, but only one copy, regardless of which parent the copy originated from. Sorry. So this is what forms the rules of chromosome inheritance normally. As you can see from the diagram, the pink and the blue bars represent the chromosomes originating from the female, which is the egg-producing organism, and the male, which is the sperm-producing organism. And at the very bottom level, if you look, you can see that there's a 50-50% chance of either having a pink or a blue chromosome. Humans pass on their genetics in this way. So this is why you probably share some characteristics with your parents, as in hair color, eye color, nose shape, or anything. Now, the odd genetics of fungus gnats, they break the rule that is normal chromosome inheritance with their bizarre genetics. Not only do fungus gnats have an alternative sex-determining system in which females alone determine the sex of their offspring, they also have no paternally inherited genetic information transmitted from the males. Therefore, they only transmit the copies that they receive from their own mothers. So you just have constantly female chromosomes being transmitted. So if we model this concept onto humans, you will only get the traits from your grandmother and your mother, and the power to determine whether a baby is male or female is only given to the mother, as opposed to how normal genetics is, where it's a mix of the X and the Y. So the female genetics holds all the powers in fungus gnats. If you look at the diagram, you can see that on the left-hand side is the female, and on the right-hand side is the male. The female has 2 pink chromosomes, and any eggs that she makes will have pink chromosomes. The male has a blue chromosome, which originated from his father, and a green chromosome, which originated from his mother. With normal genetics, they would have a 50-50% chance to get any of the color. However, as this is fungus gnats, instead, they only pass on the green chromosomes. And then in the next generation, the female, which is on the left, can make eggs with either a green or a pink chromosome, whereas the male can only pass on the pink chromosome. In my research at the Ross Lab, in the Institute of Evolutionary Biology in the University of Edinburgh, I studied fungus gnat flies, the specific species of Lycoriella ingenua, which produce mixed broods of both males and females. My research is made up of 2 experiments. The first being to investigate if sex ratio in Lycoriella ingenua is a trait that is passed on from the parent to the offspring. And then the second experiment is to test whether temperature will have an effect on the sex ratio. In particular, if there's an increase in temperature, if there'll be also an increase in the number of females. So the Ross Lab obtained the Lycoriella ingenua flies from a mushroom farm near Edinburgh. So the research that I'm doing can be directly fed back to the owners of the mushroom farm. The flies have the following life cycle. They have an egg, then they have 4 stages in larva, then they pupate, become a pupa, and they become adults. The fungus gnat larvae are the actual pests to the agriculture. Females typically lay their eggs under the soil, where incidentally plant roots and the mycelium, which is like the plant root equivalent of mushrooms, are found. And then when the larvae hatch, they basically will start eating anything in front of them. And occasionally this will be the roots of plants, which is very unfortunate. But this will also decrease the yield that is produced in the plants or the mushrooms. as females can produce up to 100 eggs each. An increase in females means that there's, in the population, basically means that the population can expand very rapidly, and then there will be more larvae in the soil, so there will be greater damages to all the crops. Here's a picture of our key players in mating. The female, which is on the right, has a thicker body and is typically larger than the male. And on the left is the male, which can be identified by having like claspers on the abdomen. For my initial experiment, I set out with the aim to see if the ratio of males to females that a female fungus gnat produces is inherited by the daughters. To test this, I had set up vials with a female and a male taken from the mass culture of flies. The flies mated, the female laid eggs, the eggs hatched, larvae matured, and then became adults. I would then count the number of males and females that had emerged to establish the sex ratio. Then we created 5 mating vials where basically siblings were crossed. And when the adults emerged from those, they were also counted. And so then we had the daughter-sex ratio, which also was established. Then an average of the 5 vials was taken and then compared to the sex ratio of the mother. I examined the data and I found that the sex ratio is indeed heritable. So if a female, for example, produces a sex ratio of males to females of 40 to 60, the daughter will produce a brood with a very similar ratio. The follow-up experiment that I'm currently in the process of running is the temperature one. So I'm seeing if there's more females produced at a higher temperature than it would be at a room temperature. In another species of fungus gnat fly, increasing the temperature led to an increase in the females produced per brood. It is important to note that the changing of the temperature of the vial filled with larvae does not actually change the sex of the larva itself, but rather changes the sex of the offspring that she will produce. Modeling my methodology off of what was done in that experiment involving the other species of fungus gnats, I set up vials from the mass culture flies, with each vial having 1 male and 1 female, as was done in the previous experiment. And this was set at 18 degrees. Then eggs were laid, the larvae hatched, they matured, and then the sex ratio was produced, and this is the F0 sex ratio. Then sibling vials were set up again, and this became the F1 generation, where the eggs were laid, the larvae emerged, and when the larvae were in larval state, the different vials were set into the different temperatures. So some were set into the high temperature, some were set into the room temperature, and some were put into the low temperature environment. Then when the pupa became adults, the F1 sex ratio could be derived. Since the sex ratio that a female produces in her offspring is determined during pupation, and the temperature is not having a direct effect on the pupa itself, this is not the sex ratio that we were actually looking for. So we had to run this whole thing again to then see if the sex ratio is indeed different within the different generations due to temperature. As you can probably see, this would have been like, we had to run the F3 generation so we could find out the sex ratio of the F2 generation. But yes. So why would I even study any of this? I guess I get asked this question quite a lot, and I really love talking about my flies. So I've had to find an answer to this question. And so there's 2 major reasons why this is cool to study. First off, for knowledge, just general knowledge. The weird genetics that fungus gnats have is very rare even in insects. Not only do they have is there a genetic effect on the sex determination where only one of the sexes, the females, pass on their chromosomes, there's also an environmental effect of temperature. Reptiles like turtles use environmental temperatures to determine the sex of the eggs. But this mechanism is unheard of in insects. In fungus gnats, temperature works together with the existing genetic mechanisms to become truly unique and to be a really cool system to study in terms of evolutionary biology. Secondly, studying fungus gnats is important because they are an economically detrimental pest. For example, in the UK, they damage the yields of mushrooms in the farms. So then there's less yield, and then so this affects the consumers, the farmers, the government, all of them. if the fungus gnat sex ratio does indeed get skewed in favor of producing more females at a higher temperature, which higher temperatures could be because of climate change, then we need to develop new strategies to control fungus gnats. Because if you remember from what I've said before, an increase in females will mean more larvae in the soil, so more damages. So studying whether temperature has an effect on Lycorellia ingenua can inform us whether mitigation strategies need to be, like new ones need to be made to basically mitigate and also eliminate fungus gnats. I would like to take a second to thank my supervisors at the University of Edinburgh, Dr. Laura Ross and Robert Baird. And thank you for listening to me, and I'll take any questions.